Yes, welcome to the new video. Compound lipids. So as you know, the lipids are classified into three major categories. One is simple lipids, second is compound lipids, and third one is called derived lipids. So we move on to the second category, that is the compound lipids. So lipids are what? Lipids are the esters of glycerol and fatty acid. They are esters of glycerol and fatty acid. And how many fatty acid are there in a lipids? There are three fatty acid. This is one glycerol molecule and three fatty acid combined together to form a lipid simplest structure. Now here what happens? One glycerol molecule is there and two fatty acid are there, and the third fatty acid position is replaced with a non-lipid. Hence it is called compound lipid or simply called the heterolipids. So what is the definition of compound lipids? The lipids that links to a non-lipids. The non-lipids are either phosphoric acid or carbohydrate. The non-lipid may be phosphoric acid or carbohydrate. Types of compound lipids. So one is the phospholipids and glycolipids. So what is the difference? In phospholipid, the third position of the fatty acid is replaced by phosphoric acid. Here, the third fatty acid is replaced by glucose or a carbohydrate, hence called glycolipids. And there are two examples. One is called lecithin and cephalin that comes under the phospholipids. Cerebrosides and gangliosides are the example for glycolipids. Now, move on to the first category of compound lipid, that is the phospholipids. So what are they? It's a compound lipids. And what is it made up of? It's made up of one glycerol molecule. How many fatty acid? Two fatty acid others. And the third position of fatty acid is replaced by what? Phosphoric acid. And there is an additional uh, structure that is also called the choline. Molecule, two fatty acid, and a phosphoric acid group is the. So what's its function? They are the structural components of membrane, which is abundant in brain and kidney. And they are also amphipathic. Amphipathic means what? They have a hydrophilic end and a hydrophobic end. There are two ends. One is water loving end and water hating end. So one of the example for lecithin is a phospholipid. They are esters of glycerol and fatty acid. There are two fatty acid, one fatty acid, two fatty acid, and the third position is occupied by a phosphate group and additional choline group. The two OH groups are esterified with the two fatty acid. So there is an OH group, here an OH group, here another OH group. There forms an ester bond with the two fatty acid. So one glycerol molecule forms two ester bond with two fatty acid. And the third position is esterified with phosphoric acid group. And there is an additional ester bond with choline. Choline is a nitrogenous base. Okay, this is the basic structure of what? Lecithin. So what is the basic structure? One glycerol molecule is esterified with two fatty acid and a phosphate group and choline. Very simple structure. So what is biological functions? Where are they found? It's found in all animals: brain, nerve, sperm, egg, yolk, and the seeds and sprouts of the plants. What's this color? It's an yellowish gray solid in color. They are required for normal transport and utilization of other lipid in the liver. Lecithin is required for the transport of other lipids in the liver. Lecithin on hydrolysis yield choline and two molecules of fatty acid. On partial hydrolysis by lecithinase, a venom. Lecithin on partial hydrolysis by the enzyme lecithinase. One fatty acid is removed. So when it contacts with the blood, the RBC undergoes hemolysis. So this is what is a viper. Is poison is hemotoxic, and this venom contain what? An enzyme called lecithinase. So this lecithinase lecithin to form lysolecithin. So that results in what? Blood hemolysis. So this is the biological functions of lecithin. Is a another function is what they are surface active agent, helps in emulsification of fats. You heard about that emulsification of fats. Emulsification simply means breaking of complex fat into simpler molecules. So this process is called what? Emulsifications. Now there is a important lecithin is dipalmitoyl lecithin is a lung surfactant which helps to lower the surface tension, which is essential to prevent the collapse of lung alveoli. The alveoli is just like a small balloon-like structure or bunch of grapes. So it need to be expanded for the effective function of the respiration. If it collapses, the air cannot enter. So in order to prevent the collapsing of this lung alveoli, the surface tension around the alveoli should be reduced. That is by the help of what? Lecithin. So dipalmitoyl lecithin is present above the on the surface of the lung alveoli. The lung alveoli is covered by a thin layer of what? Dipalmitoyl lecithin, which is to reduce the surface tension in order to prevent the collapse. So another examples for phospholipid is cephalin. Cephalin, the same structure. This is the glycerol molecules is esterified with the two fatty acid. The third position is esterified with a phosphate, and instead of choline, it is ethanolamine. So this is the structure of what? Ethanolamine. So what is the difference? The difference is here. In lecithin, it is here the phosphate is bonded with, here it is in cephalin, it is esterified with ethanolamine. So it's also known as what? Phosphatidyl ethanolamine. So how many commons are there? There are four commons: glycerol, one glycerol molecule, two fatty acid, one phosphoric acid, and one ethanolamine. The important fatty acids are that normally uric acid, oleic acid, linoleic acid, arachidonic acid. There are two types of cephalin: alpha phosphatidyl ethanolamine and alpha phosphatidyl serine. So based on the groups attached with the phosphate, there are two types of cephalin. One is called alpha phosphatidyl ethanolamine, that is this picture, and alpha phosphatidyl serine. So this is a serine amino acid linked here. This is a serine. This one is a serine which is linked to the phosphoric acid or phosphate group. So this is the ethanolamine linked to the phosphate group. So there are two types of cephalin: alpha phosphatidyl ethanolamine and alpha phosphatidylserine. Hence, with a less than a split of the fatty acid from the cephalin, leaving hemolytic lysocephalin. It causes hemolysis of RBC. It occurs in animal tissue, brain, soybean oil, etc. The major role is in the organization of protoplasm. So these are the major functions of phospholipids. They form the cell membrane, transport of triglycerides in the form of chylomicron. You heard about that. It helps in blood clotting. It prevents accumulation of fats in the liver. So it prevents fatty liver. Phospholipids in bile makes cholesterol soluble, and it acts as a lung surfactant. So that's all from phospholipids.